Hello, everyone. I am so sorry we're a few minutes late. We had some technical difficulties, but we are hopefully all here and ready to go. My name's Pip Cohen. I work for World Fish, and I'm lucky enough to have been part of this collaboration that today is launching FishNutrients, which is an innovative new tool for research and policy that aims to reduce malnutrition. To set the scene, let me share with you a very short video that shows you who this collaboration involves and what we've done.
Fish are rich in multiple micronutrients, essential fatty acids, and protein, providing a powerful contribution to diets, particularly for vulnerable and marginalized peoples.
But fish are not made nutritionally equal.
We know this because for years we've been compiling quality-controlled nutrient profiles for different foods, including for fish.
But data were missing for hundreds of fish species in many regions, particularly in Africa and Asia, where diets are most dependent on fish.
And measuring the nutrient composition in fish is difficult and expensive.
So, we joined forces.
Across organizations.
Across databases.
And across nations.
And we developed a statistical model, which means that we can now predict the nutrient profiles of every species of fish, both marine and freshwater.
To build it, we combined nutrient data from the open access data platform Infood.
With ecological and environmental data from FishBase, the world's largest encyclopedia of fish.
This means that for the first time and for every species of fish, you can find the micronutrients, omega-3 and protein concentration on FishBase alongside the primary data.
We used this model to find where there were injustices in global food systems and what to do about it.
Our scientific papers have highlighted where fish catches can help address malnutrition.
We've generated new understanding of how foreign fishing and trade move essential nutrients from fish between nations.
We've looked at how climate change affects the nutrients available from fisheries.
And which geographies and species are most vulnerable to shocks.
Connecting this data and building these models into FishBase has allowed us to further develop FishBase's functionality.
Now, everyone with an interest in fish and nutrition can use this information too.
So visit FishNutrients in FishBase today for any fish species or region and download the data you need to test hypotheses and develop evidence-based policies and programs.
Over to you.
Thanks again, everyone, for joining us. As I said before, I'm Pip Cohen and I'll be moderating this launch of FishNutrients. We have received some requests to record the session so that we can make it available to those that can't be here right now. Through the event, please do feel free to ask questions in the chat. We hope we have time to answer these live as we go, but if we do run out of time, as you know sometimes happens in these things, then we can try to get back to you directly or use your questions to help us develop further information for everybody. We have a really impressive lineup of panelists who have all been really key collaborators in this initiative. So let me introduce them to you. Professor Daniel Pauly is the principal investigator of the Sea Around Us initiative at the University of British Columbia. He's authored over 1,000 articles on fisheries and related topics. He is now the world's most cited fishery scientist. As the co-creator of FishBase 30 years ago, we are thrilled that Daniel is a champion of this collaboration and what we've now developed. Dr. Shakuntala Thielstedt is World Fish's global lead for nutrition and public health and the recipient of the 2021 World Food Prize Award, which recognises her 4 decades of work to improve nutrition and health for millions of malnourished children and their mothers across Asia and Africa. Shakuntala has mentored many contributors, including me, to this work, and has long championed the need to consider the nutrient qualities of food, and in particular, fish that she refers to as a superfood because it's rich in bioavailable micronutrients, fatty acids, and protein. Professor Christina Hicks is within the political ecology group at Lancaster University. She studies how social, ecological, and institutional settings shape outcomes across aquatic and coastal systems. The work that we discussed today was spearheaded by Christina more than 6 years ago and has since brought us better understanding of the macro level patterns of fish micronutrient distribution. Associate Professor Aaron McNeil is Canada Research Chair in Fisheries Ecology at the Ocean Frontier Institute, Department of Biology at Dalhousie University, where he leads the Integrated Fisheries Lab and which researches small-scale fisheries across diverse contexts. He has incredible skills in Bayesian modelling that this collaboration has relied on immensely. Dr. Maria Deng Palomares is co-creator and lead of Sea Life Base, which is the younger sister of FishBase. She's the manager of Sea Around Us initiative at UBC. She's the consortium director for FishBase and the science director of quantitative aquatics. Deng has been with FishBase since its inception more than 30 years ago and has intimate knowledge of what lies behind FishBase, so the numbers and the coding that we too rarely think about. Dr. Kendra Travail leads research and advocacy work to end overfishing and to promote sustainable, responsible and equitable fisheries. She works for Minderoo Foundation's Flourishing Oceans initiative and has a growing interest in the food and nutrition security wins that can be brought about by reducing overfishing and improving fisheries governance, especially towards ocean equity goals. Dalhousie University and the Ocean Frontier Institute have been kind enough to host today's launch, and to welcome us, I'd like to welcome CEO and Science Director, Dr. Anya White. Anya.
Thank you very much, Pip, and thanks to all of you who've dedicated so much time to creating something that will make such a difference in the global community. The tool we're launching today has been developed thanks to the breakthrough paper led by Christina Hicks and a number of our panelists today, a paper that was published in Nature in 2019. And this describes the substantial potential that lies within the world's capture fisheries to address micronutrient deficiencies in coastal populations. And that's these small things like vitamins and minerals, as opposed to the big nutrients like nitrogen and phosphorus. Now, thanks to this research, we now have a much more accurate way to determine nutrient data in fish. And this new information will help advise nutrition-sensitive management of global and local fisheries to improve the predictive ability and management use, particularly in developing nations. We at the Ocean Frontier Institute are very pleased to host this event and support this research, as our mandate is to help inform international policy. I would like to congratulate all of you who have dedicated yourselves to this cause and invite those of you watching the webinar to do what you can to share the news and help this launch become a true success. Now, let me hand over to our guest, Daniel Pauly.
Thank you very much, Anya. The information system used to make the macronutrient data in fish available to the world is FishBase. Now, FishBase is a freely accessible database and website with a vast amount of information in all freshwater fish and marine fish of the world, over 34,000 species. The development of FishBase was initiated in the Philippines when there was no internet, no Google, no Wikipedia, and scientists and other persons requiring information on fish could not get any information without significant money and time investment. FishBase was created to meet this need for information, not by providing list of reference and bibliographies, as was the rule then, but by encoding the information itself and documenting its source. This approach was highly successful, and FishBase is now used all over the world, notably in all institutions of higher learning with teaching and research program involving fish. In the process, generating about 900,000 page views per month, by 500,000 users, soon 1 million. FishBase also garner about 1,200 citations per month in the scientific literature, which is more than most scientists garnered in their entire career. We live in the age of big data in genomic and cosmology. However, we must remember that this age was ushered by databases such as FishBase, which assemble the content of more than 1,000 books and can be updated as required, as book cannot. Because information encoded in FishBase contains a wide variety of source and emphasized data from developing countries, big data, as represented in FishBase, allows researchers all over the world to undertake wide-ranging analysis, as is the case for macronutrient in fish. Moreover, FishBase and its component, including the new data on fish micronutrients, are a public good, similar to Wikipedia, which makes knowledge freely available to the world and help advance solutions to various fish-related issues, ranging from fisheries management to food security issues. Thus, in the name of all creators of FishBase, and there are many, I must say that I'm delighted to see this macronutrient database, that it has been incorporated into FishBase, and this will make it even more useful than before. Very pleased to hand over to you, Shakuntala. Shakuntala?
Thank you, Daniel. After very many years of research in fish nutrient composition, I am so happy that we now have a shift in emphasis from feeding people to nourishing nations. This is a critical shift in the way we think about food systems, a transition from quantity to quality. The collaboration has brought this transition to the fisheries research and governance communities by shining a light on the quality of fish as a rich source of multiple bioavailable micronutrients, essential fatty acids, and protein. This is a huge step forward that now needs to be followed by holistic shifts towards nutrition-sensitive fisheries governance. And with that time, I do hope that this will also include another aspect of food quality, that is food safety. This collaboration that led to the inclusion of FishNutrients in FishBase has amplified the incredible resources made available by InFoods, a food composition database coordinated by the FAO. In looking at the empirical data on fish collated over the years by InFoods, this team found huge data gaps in the diverse fish species which we all know are eaten by people across the world. So this collaboration has gone one step further and filled those data gaps with estimates from novel predictive models. This collaboration brings to the research community a powerful new tool to understand the potential of diverse fish species and fisheries to address malnutrition and nourish people and planet. These fish nutrients brings to the world of human nutrition and public health, brings the world of human nutrition and public health together with fisheries. The challenge for us as researchers and educators is to embed these tools of fisheries managers and health professionals. This data and knowledge can now reach all the way to fisheries governance policymakers, investors, and also to the professionals who develop national food-based dietary guidelines to include fish. Let us celebrate this achievement of FishNutrients. However, let us also continue this work and expand it so that we also take into consideration the huge diversity of all aquatic foods animal, plants, and micronutrients that are consumed around the world. Thank you, and over to Christina.
Thank you very much, Shakuntala, and everyone for joining us today. Our nutrition collaboration started quite modestly about 7 years ago with what we thought were two simple questions. How nutritious are global fish catches, and can they help address malnutrition? As we started gathering data on the nutrient content of different species of fish, two things became quickly apparent. Firstly, that fish are not made nutritionally equal. So some species, including small pelagics, are rich in a number of micronutrients, whereas others not so much, which really highlights the importance of understanding the nutrient composition of the species that we eat. And second, there were a lot of gaps in the data. Unsurprisingly, a lot of information existed on commercially important species that are consumed in the West and on protein, whereas there was far less information for species found in the tropics and for some of the micronutrients that are critical to physical and mental development. So we set out to see if we could plug some of these gaps by drawing on the information that was available, so for over 500 different species. And we initially focused on 7 nutrients, including vitamins, minerals, fatty acids, and protein. And FishBase was the natural place for us to turn for the types of information that we needed that could help us explain the patterns of nutrient composition in fishes. So it's now particularly nice, as Pip highlighted at the start, that we can return the favour and close the circle by connecting our model and data to FishBase. Now, in a moment, Aaron will speak in more detail about the model that we've used to plug these gaps in nutrient composition. This model did allow us to answer our initial questions. And in our analysis that we published in Nature, we found that catches from many countries' EEZs contained sufficient quantities of nutrients to close nutrient gaps in local diets, and in doing so address malnutrition, but only if a greater portion of the catch could be retained and consumed locally. In addition to these initial questions, this work in the model has opened up a whole new area of research that allows us to ask a range of theoretical questions, applied questions, and also support the development of evidence-based policy for nutrition-sensitive fisheries management. For example, Kirsty Nash is leading work to map the nutrient flows through foreign fishing and trade. Ava Mayer has read work exploring the vulnerability of nutrient-dense species to fisheries and also to climate change. William Chung is leading work to understand how climate change has and will affect the nutrients available from fisheries. And James Robinson has led work to really try and understand the mechanisms through which these climate-induced changes occur. So we're really happy that now you can also access and download the empirical or model data for individual species of fish, or for full lists of species. For example, if you work in a particular geography, such as Kenya, you can download the full list of species found in her waters, together with the nutrient concentrations for each of these species in approximately 30 seconds. So we really hope that you will be able to use this resource to answer your own questions at the intersection of fisheries and nutrition, or develop policy. I'd like to now hand over to Aaron to talk a little bit more about the model and the developments there. Thank you.
Thanks, Christina. So, one of the major limitations for people considering fisheries from a nutrient perspective has been data. So in general, sampling has been from only a few key species in a few locations, often in high-income countries, as Christina stated, but also from a few low-income nations where people have active food research programs. So to overcome this limitation, we developed a statistical model that used information from the fish species that we had observed to estimate the nutrient content of species that we hadn't. And so the models use key attributes of fish relatedness and traits to establish relationships between these observed and unobserved species. So fish relatedness here refers to their phylogeny, the shared genetic history of those species. And for traits, we're referring to factors related to nutrient pathways in fish diets, the temperatures and environments that they inhabit, as well as the size and growth of each species, all factors known to affect their tissue composition. So in testing out our model on new data collected by our colleague James Robinson in Seychelles, which is a tropical area that was quite distinct from the set of data that we used in our original observations, we've been pleasantly surprised at how well the model has been performing in predicting the nutrient content of new species. These new observations were within our 95% predictive intervals between 68 to 98% of the time, depending on the nutrient. So while these results are very encouraging, better than I would have expected given the difficulty of making out of sample predictions in ecology, we know that the model will be inaccurate in some cases. So some initial work done with our colleagues suggests that spatial variation may be important for some nutrients, where areas of high runoff, for example, affect concentration of things like iron relative to, say, low island areas. So while we're thrilled to see our results integrated into FishBase, our work really is only beginning, and the more data that can be collected from a wider variety of species, the better our model predictions will become. And other researchers may have new ideas about how the model itself can be improved. So I hope that this launch with FishBase is only the start of an iterative process that will make our estimates as accurate as possible. And to that end, all of our models and code are available through a GitHub link on FishBase. And I'd encourage anybody with data or model structure ideas to reach out to any of us that we can integrate that new information. So a little bit about FishBase. I'll hand you over to Deng Palomares.
Oh, thank you so much, Aaron. As Pip said, my role in this initiative was to ensure that the data can speak to users in ways that make sense on the FishBase platform. Now, out of the hundreds of thousands of people who use FishBase every month, as Daniel has mentioned, only very few people have seen what lies behind FishBase. So I can give a peek now as I explain a little about how we brought this nutrient data into FishBase. So the FishNutrients tool was built in Quantitative Aquatics, the Philippine NGO that hosts FishBase and SeaLifeBase. It was built by the IT team that is led by Ms. Josephine Barile and the project's software engineer, Ms. Camille Abay. To start with, we used the structure of an existing table in FishBase that contains data on the chemical composition of different fish body parts. That is data on moisture, protein, fat, and ash that these different fish body parts contain. There are similarities in the structure of the new nutrient data with existing FishBase tables. And this made the incorporation of data from this project a little bit more straightforward. So all of the different components of the FishNutrients database were easily brought up to the global data standards that FishBase adheres to. Once the interface was built, the interface that you will see when you search nutrients data in FishBase, we then consulted with Aaron, Christina, and Pip and their teams on how users will most likely interact with the interface. This consultation and several rounds of quality assurance tests resulted in the tool as you saw in Christina's slide, and which is being launched today. The nutrient data for 500 species and modeled estimates for over 5,000 fish species could be integrated within a relatively short period of time. Behind the scenes, as the model developed by Christina, Aaron, and their teams, uses pre-processed fish life history traits data, as Aaron had mentioned, a mechanism to collate this data from FishBase in the format necessary for input into the algorithm explained by Aaron is now in place and will be used to update the FishNutrients page on a regular basis. This assures continued availability of updated FishNutrients data via FishBase. The model published by Christina, Aaron, and their teams couldn't have been built if it wasn't for the large data set managed through FishBase. And this initiative is a great demonstration of how established open access data infrastructure can make data sets, such as the FishNutrients data, freely available via the World Wide Web. The panel that we have here today is a great celebration of data commons which can often be taken for granted. Thank you for everybody. Now let me hand over to Kendra.
Thank you, Dang. As Pip mentioned, I've had the pleasure of coordinating a grant from Minderoo Foundation that has enabled this inspiring group to take this important step in a long collaborative journey. Our goal at Minderoo is to transition the ocean to a flourishing state by 2030 by accelerating progress towards sustainable, responsible, and equitable fisheries. To us, this new tool reflects an important intersection between SDG 14, Life Below Water, and SDG 2, Zero Hunger, highlighting the role of fisheries in meeting global food and nutrition needs. Minderoo is one of many funders who see immense value in platforms like FishBase, Sea Life Base, and the FAO InFoods, which support the data commons and serve up information to policymakers and researchers around the world. In supporting the development and launch of FishNutrients, we are honored to stand side by side with other funders, including the European Commission, CGIAR, Oak Foundation, Australia's Fisheries Research and Development Corporation, and others. We recognize the launch of FishNutrients as a key first step towards aligning fisheries sustainability and health and nutrition goals. And we're excited to continue working in partnership with the research team to ensure the best and most current nutritional data is available for use. To that end, I'm excited to announce Minderoo will be funding the first annual Fish Nutrition Data Sprint, focused on incorporating new empirical data into the model and improving the nutritional value estimates generated across species. These data sprints will run over 4 weeks, with the first sprint starting the 1st of May 2022 and culminating on May 28th to coincide with World Nutrition Day. In addition to adding more data and improving model estimates, these sprints will be used to raise the profile of the value of fisheries, fish, and other aquatic foods for human health and highlight the value of this data for generating knowledge about the role and value of fisheries in meeting food and nutrition security targets. I'm thrilled to see a continuation of this multidisciplinary cross-organization collaboration and can't wait to see where it goes from here. Pip, I think we're back to you.
Great. Thank you, Kendra, and thank you, everybody. We are really excited and really grateful for the support of Minderoo and the other donors that Kendra mentioned. We're particularly excited that Minderoo are supporting this, one of many, we hope, FishNutrients data sprints. So as Aaron and Dan were saying, these sprints will allow new published data to be incorporated and made visible through this site, and also refresh the model, which improves its accuracy, the accuracy of its estimates, so it learns and builds accuracy. So this launch and this collaboration and your interest in this tool and all that you are able to do in this space is really timely, because we're now in the decade of ocean science and in the decade of action on nutrition. Next year is the International Year of Artisanal Fisheries and Aquaculture. So during this year, 2022, we're really keen to see how this work can illuminate and properly recognise the contribution that small-scale fisheries in particular make to food and nutrition security globally. And so, well, this is a great milestone, and we're really happy to celebrate it and celebrate it with you. It is also just the start of many other journeys at the nexus of these 2 decades and their associated calls to action. We do have some moments to answer some questions. I think the team have been trying to answer questions as they roll in. But I saw one question. I might start with Prana from India, who had a question. Is there any nutritional composition difference between the same fish species from different geographical regions? And the answer is there is difference. It matters where the fish came from. And so for that reason, when you look at FishBase, you can find the geographic region where the empirical measures came from. And so it's really important to kind of look at those. And of course, then that geographical variation builds in a range of uncertainty in the model predictions. I saw another question from Yeppie Colding. Hi, Yeppie, really glad that you're here. And it reflected some other questions. So Yeppie, let me find your question. And I'm going to throw this to Christina. So Christina, get ready to catch. Depending on the size of the fish, then not all the parts are eaten. The nutrition composition is not homogeneous in the fish across the whole animal. Other nutrient data that we have made available through FishBase, are they referring to the whole fish, such as small fish eaten whole, or to parts normally eaten, such as fillets?
Thank you, Pip, and thanks, Yeppie. That's a really important question. I started typing it, so I'll send the answer so you'll have that as well. But yeah, absolutely, you highlight a really important point that particularly in certain geographies, particularly for small species like small pelagics, the fish are often eaten whole. And so the nutrient content will vary. I guess the short answer is if you go to FishBase and you look at the empirical data that comes from the literature, all of that information will be there. So you will be able to see what body part the nutrient composition refers to. It's generally either tissue, a lot of it is tissue, but some of it is whole, particularly for smaller species, and some of it is whole minus parts. For the modeled estimates, and Aaron can speak more to this, those parts that have been used in the analysis are included in the model. So the model estimates tells you tissue concentrations for tissues, but it accounts for the fact that some of the values that have informed that have been taken from the whole fish and adjust for that accordingly. So Aaron can perhaps pick up on that in terms of the modelling. One thing I did just want to add, I think, Pip, in response to the previous question, is how nutrient content varies by geography is an area of ongoing research. So we know various factors do affect the nutrient composition of different nutrients. So omega-3 tends to be lower where temperatures rise. We've got Ava Mayer's currently conducting research trying to understand what are the geographical patterns of micronutrient concentration in a range of different species. So I think, like a lot of these things, there's a lot of questions to ask and a lot that we still need to find out. And that relates also, Yeppie, to your question around how does the content vary for different species. So hopefully we'll plug these gaps going into the future. Thank you.
Yeah, briefly, just to echo Christina's comments, both of these comments really speak to data and the fact that we know that these things will vary, but that we need the data available that across geographies, for example, that will allow us to build covariates into our model that will help with the predictions into places that may not have observations. So the more data we get in, the more contributions about how these things vary, the better we're going to be able to do.
Great, thanks both. Another question from Ghislaine Llewellyn. And Shakutala, this is for you. You got me? All right, is bigger better when it comes to the diversity and availability of micronutrients and fish size? Or rather, is it that best things come in small packages?
Depends. Because remember, not very many people eat fish on its own. Fish is part of the meal. It's one of the what we hope diverse foods on a plate of other foods. And in many countries where we work in Asia and in Africa, we see that fish is cooked with other foods. So it has the power of bringing with it other nutritious foods such as vegetables and spices. And then again, it's eaten on a plate with other foods. If you go to Bangladesh, there's hardly a plate of food, especially for women, that does not also contain green chilies and lemons, which are essential for supplying micronutrients and enhancing the bioavailability of nutrients. Thank you, Pip.
Great, thank you. Another question here from Mike Ackerster. Hi, Mike Ackerster. This one, Aaron, this is a good one to link back to the modelling. Presumably these questions are moving around. Presumably the food that the fish eats influences its nutrient composition. Can you answer that one, Aaron?
Yeah, certainly. And that's, in our original model, we had sort of different, we thought pretty hard with Nick Graham, our colleague, and others about how nutrients flow through food webs. How do you end up with nutrient content that you're eating? And of course, it has to do with your trophic position and things like that in the food web. It has to do with what's available in the environment. And then it has to do with, those interrelationships as well. And so we tried to put as many of those together, thermal regime, benthic versus pelagic, these sorts of characteristics, and all of those things contribute more or less to different nutrients. So if we go back to Christina's paper, there's a graphic there that shows the sort of estimated effect sizes so that you can see how relatively important these things are. And we have another new paper with a slightly updated model, which is what underpins the estimates here, that will have revised estimates for those things. So, the most obvious ones are things like omega-3s, which are quite dominant in polar regions. So you see them just based on the food web. It's what is really interesting going forward is to see, again, the spatial variation. So, there's a species of mullet that is everywhere, and freshwater, brackish water, marine, all over the place, a very cosmopolitan species. And so looking at that species and seeing how the nutrient content varies in that particular species, for example, will be quite interesting. So all of those things are really important. And again, those aspects of the local food web, because ultimately it's the phytoplankton producing these nutrients that then get amplified up into the food web. Seeing how those things vary in space is going to be really important. So it's a great question.
Great, thanks, Aaron. Daniel, can you check the message box, because I've put a tough one in there for you, and you might need to prepare. Aaron, just while I've got you, do you share the level of confidence or accuracy around the predicted data?
Yeah, so a lot of this is uncertain, right? We're in new territory, and so the types of models that we built are Bayesian, and then what that means is that it represents our current best state of knowledge. So while, predicting this many species from a subset is quite perilous, I would say, in general in ecology, we've done pretty well. And more to the point, I think we've established a beachhead to then make better and better predictions as we go forward. So as I alluded to before, there's places where these things will be inaccurate. And we want to know about those because that's how we can learn to improve these estimates. But for anything that we've estimated, because it's a Bayesian model, there's a probability distribution that describes our uncertainty about any one of these aspects. And so for some attributes, things are more certain, and for others, it's more uncertain. And that's just the nature of the state of knowledge that we have.
Great, thanks. Daniel, I'll give you a few more moments. I'll go to Deng. on a question that a couple of people have asked about, yeah, I mean, we recognise that this effort is at the moment limited to finfish. And Deng, I'd love some thoughts, I guess, about what opportunity does maybe marrying up the data that are housed by InFoods with a database such as Sea Life Base offer. Is that possible?
Yes, that's possible. The Sea Life Base uses the same platform as FishBase, so it's very easily the nutrients database here and the interface and the structure behind it is very easily copyable into Sea Life Base if data exists. So the thing would be to check what kind of information needs to go into the algorithm and make sure that for the species with nutrients data, we have all of those information filled up. So yes, it is.
Thanks, Deng. And just to give a shout out to our partners at FAO InFoods, who are the data commons that house all of the nutrient composition data for all foods. And someone else was asking about different products of fish, so cooked fish, tinned fish. That is the place to go. There's been an additional effort InFoods Plus to focus on aquatic foods that is underway, and hopefully that will be revealed and made available as well shortly. So Daniel, I posted a very tough question in the box for you.
The chat box? The chat?
Yes. Oh, sorry, you haven't had time to prepare. The question is, are there any suggestions on how we can accelerate the use of these data into better fisheries and nutrition and health policies? So the challenge, how do we make sure that these data aren't just swimming around in the echo chamber of scientists and move into shaping policy? The
group around Christina had a huge megaphone with a paper in Nature, and usually these papers are not overlooked. Now it is our job now to make people know that FishBase has the follow-up story or the follow-up data, and FishBase is available in many places of the world. The use is superficial. People often use it only to know the name of the fish because they have a fish or something, and lots of people don't know about the depth that it has, and that's nutrients are part of the depth. I think it will be our job in all our institutions in our project to make people aware that there is this magnificent addition to FishBase, and given the distribution, the wide use of FishBase, and the readership of Nature, the intersect of these two things should lead to a pickup by. Also, FAO is likely to take it on because FAO is a member of the FishBase Consortium, and as part of the FishBase Consortium, we will disseminate this also. We have a meeting coming in Paris, celebrating 30 years of FishBase plus one COVID year, and we will talk about this new things that are in FishBase, and we will also in the next year include that in the sea rounders. As a product, the sea rounders also has the data are widely used. They basically we will incorporate nutrients as a currency in a catch. So the catch can be expressed in tons, it can be expressed in dollars, but it also can be expressed in nutrients. And when we do that, I guess that is going to be an opening up another avenue because lots of people use the sea rounders data. So that's what we'll do.
Great, thank you, Daniel. Shakuntala, more thoughts on how we shift this discussion from one amongst researchers to the science policy nexus?
I'd like to talk about the policy on one side, but I also want to talk about the broad usage on the other side. You and I have talked a lot about that this kind of work and this kind of research must be made into tutorials, into course components that can be offered at universities and embedded into curricula. That's such an easy and good way to pass it on to the next generation of researchers and implementers. And from where I stand, and I look at the developed world, how they use nutrient composition data, for example, in national food-based dietary guidelines, especially now in COVID-19, if you have to do rations for the poor and vulnerable, make up feeding programs for school feeding children, you need national food-based dietary guidelines, and you can't have fish and other aquatic foods within those guidelines if you do not have the nutrient composition data. So this is all important for policymakers and also for passing on to the other generations in the work that we do. Thank you.
Great. Thank you, Shakuntala, for that. We'll have one last question. There are a few questions around fish feeds, and particularly feeds based on plants and algae. I should clarify that we focused the samples that we shared on those fish that were wild caught. But nonetheless, this is a really important area for discussion. What a feed eats, as we heard before, is what a fish eats will influence its nutritional composition. So for aquaculture, this is an important question as well. So Anya, do you mind sharing some insights on that?
Yeah, thanks, Pip. I noticed there were a lot of questions around how do we feed the fish that we need in some aquaculture applications. And I just wanted to encourage aquaculturalists to think about the new plant-based feeds that are so much more sustainable and in some ways more nutritionally valid than some of the earlier work. So Stephanie Colombo, for example, at the Ocean Frontier Institute is working on macroalgae and microalgae based fish food, which gets around the need of feeding fish to fish and makes the whole system much more sustainable. So happy to share insights on that work. And I think this is work that would be potentially transferable to local communities. So that might be a conversation that we could open up. Be very interesting.
Great, thank you. According to my time, which is 1 a.m. in Tasmania, we are almost out of time, and so I will wrap up, and we will copy those questions, and they will be useful for the work we do. I will just note one of the questions was about can we give a tour of how you navigate these new data and how they're available, where you get to them, what's what. And that is something we'd like to do in the next couple of weeks, do a very short tutorial on where you find these data and what they mean. So we can get to that and share it. So as I said, while this is a really important milestone and we're really happy and somewhat exhausted to be here today and to celebrate this with you all, it is just the start of a lot of work. And so whilst we're working directly on various ways to generate new insights and actions, including at that science policy network towards improved nutrition and sustainable fisheries, we're also really excited about what you do next and what these data in this format enable you to do and the insights that they generate. So we can do this together, continue innovation through collaboration. We encourage you to access these species-specific nutrient data through FishBase and use it in a variety of ways. contribute to the sprint. If you're in the position where you're able to do nutrient analysis on different fish species or other aquatic life, then do it to the best, the standards that have been published, and publish your data so that these can be integrated into data commons like this and like in foods. And also send us feedback, share how you have used these data, share any problems that you're finding with the interface, any feedback, as Aaron was saying, on the model, its performance, where it's performing well or where it's not performing well. All of that is really useful to make this a much more useful tool for everybody. We've posted some links in the chat box, including some email addresses. If you promise not to bombard them with too many emails, we can answer some questions directly, or we can put on other information products like that tutorial I mentioned. So lots to do, but for now, thank you so much for joining. Wishing you all a really wonderful evening or day. Stay safe, and thank you so much.